Okay, after interphase finishes, mitosis starts. First part of mitosis is called prophase. So in prophase, the nucleus begins to dissipate, all that good stuff. Clear envelope disappears. And the mitotic spindles, which are shot up by the centrioles, those are the microtubules that the motor proteins will walk on. Those eventually start to form. But also the chromatin, which is duplicated, starts to condense into chromosomes. So like this is a chromosome. Our body has 46 chromosomes, 23 pairs, and they contain all of our genetic information. So all the DNA in our bodies on these chromosomes. And each chromosome has like these little things called a gene locus, like this pink spot right here. That is a gene locus. It's a specific place on a chromosome where the gene is located. So we could, they'll have obviously more than one. So we could say this is like the gene for hair color. This is like the gene for body odor. And this is like the gene for earlobes attached or free. I don't know, it can be anything. But yeah, that's basically the place where the genes in the specific genetic location for the gene is located. They could have different alleles, like this could be like big H for, or big E for attached earlobes, or it could be little e for free earlobes, or whatever. It could be anything. But since all the chromosomes are duplicated in the S phase, there should be 2 of each of them. And they when they are duplicated, they're no longer each separate chromosomes. They are called sister chromatids. So this thing and this thing, they're both sister chromatids. And they're just the original chromosome right here and the duplicated chromosome that's duplicated in the S phase of interphase. And they're attached at this thing called the centromere. So it's like this, I don't know if you can see, it's like that black thing right here. We'll pretend they're attached. And we'll pretend that's like a little waist. It's like the waist. It's the spot where the 2 sister chromatids are attached. But yeah. Anyway, so that's prophase. After prophase, it comes metaphase. So let me see if I can demonstrate that. Let's say this is one end of the cell and this is the other end. We're going to say these chromosomes, they're lined up by their centromeres, 2 sister chromatids in each one, lined up by their centromeres. And they're going to like line up like this, all in a line. They're probably going to be off camera, and they're not in line. But they're all going to line up in a straight line in the middle of the cell. And then, here, I'm going to move the camera a little bit. They're all going to line up in a straight line in the middle of the cell. And then the let's say these are the centrosomes right here. They're going to shoot out stuff called the mitotic spindle, which is these little spindle fibers, microtubules, that'll come out here, and they'll grab onto these things called kinetochores, where they'd basically be little hooks on the edge of the centromere, or not, yeah, on the edge of the centromere. There's 2 kinds of microtubules from the mitotic spindle. The kinetochore ones, which will like attach onto the kinetochores, and they'll move these things, all these, to the middle, with the help of the motor proteins. And then there's the non-kinetochore ones, which are basically responsible for elongating the cell. Just trying to make sure my phone doesn't fall off the desk right now, but yeah. Anyway, it's about to fall. After this is the metaphase. You can see all the chromosomes, the sister chromatids attached at the centromeres, all in a line. Mitotic spindle shoots out. Motor proteins walk on the microtubules. They grab the sister chromatids and they're going to separate them. So they're going to go. Each sister chromatid is going to go to each part of the cell, and that's called anaphase. When the motor proteins come along, grab onto the kinetochores and rip the sister chromatids apart. So now you can see, instead of the 4 chromosomes, we now have 8 separate chromosomes because there are 8 centromeres out there. And yeah, so that's anaphase. You can see there are all the different poles. And then after anaphase comes telophase. Telophase, in animal cells, the cleavage starts to form. So it would be like right here. And the cells start to divide in half. They're elongated by the non-kinetochore microtubules. The daughter nuclei begin to form. And basically, they're going to start to split apart. And then eventually there's going to be something called cytokinesis. And cytokinesis is going to completely like split the cell in half. So then you've got 2 genetically identical cells, and they would both look exactly like the original cell. So this cell and this cell are genetically identical, and they look exactly like their original cell. So that's mitosis.